Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hello, everyone, and welcome back to the Genetics Podcast. I'm really excited to be here today with Dr. Ellen Reisinger, who's professor for gene therapy for hearing impairment at the University of Tübingen, where her work focuses on the molecular mechanism of hearing, and she's done some foundational work in development of gene therapies that treat hearing loss and deafness, in particular understanding otoferlin and some of the other genes that you've seen in the news lately and probably heard me talk about on this podcast. So some really exciting breakthroughs across the last few years in treating this form of hereditary deafness. We're going to talk about the underpinnings of the field and some of Dr. Reisinger's work, but also what the big challenges are today and what needs to be unblocked to make the kind of breakthroughs that we've seen more accessible to everybody with all forms of hearing loss. So we got a tall order in front of us, but I'm really excited to have Dr. Reisinger here with us today. So thank you for joining us.
Hi, thank you for the invitation.
I'd love to start with how you got into this field in the beginning. The inner ear is an amazing and beautiful organ that I remember learning about in school and just being fascinated with how we can get such fidelity from the world around us and make sense of it. What brought you into wanting to understand more and then understand when it goes wrong and what we can do to fix it?
Oh, that was partially by coincidence. When I looked for a diploma thesis, I was invited to start working on the inner ear, and I found this really fascinating. And I did molecular biology on rodent inner ears kind of now for almost 20 years now.
Did you initially get interested in therapeutics, or did you make your way to therapeutics via the fundamental biology?
I did really basic research. I studied which genes are upregulated during development of the inner ear. Then I focused more on the function of otoferlin. I did really a lot of basic research on the function of otoferlin. And yeah, there was not so much focus on therapy for quite some time. So we tried, but first attempts failed. But then I had a very talented student and she wanted to learn cloning, and she had done her bachelor thesis already in my lab. And then I decided, oh, what about this dual AAV project? We need a lot of cloning for this. Maybe you want to learn this. And then she did it. She cloned the plasmids for this project, and she did the gene therapy in the mouse models then later on. This is how basically I went into the therapeutics field.
Could you explain how dual AAV works for people who aren't familiar with it?
Yeah, right, sure. So the adenoassociated viruses, the AAVs, they are preferred tools for gene therapy because they're non-pathogenic, they are the least inflammatory, and they are very small, so they can also diffuse through extracellular fluids very nicely. They have a lot of different natural serotypes, and by choosing the serotypes, so the surface protein, you can target them to specific cells. So everybody wants to use these AAVs for gene therapy, but the problem is they can transport only a limited amount of cargo, so only rather small genes. So as you might know, you need a promoter sequence and a polyadenylation sequences for stabilizing the mRNA in the cell. So in total, you can transport genes or coding sequences of around 3,000 bases. And OTOF gene, the coding sequence is 6,000 bases. And to overcome this limitation, we split the coding sequence of otoferlin into two AAVs, and we co-inject these two AAVs, and they have the intrinsic capability of forming head-to-tail multimers in the nuclei of the target cells. And we use this intrinsic feature to bring those two parts of the coding sequence together.
Is this pretty commonly used across all of gene therapy today? Is this the first time it's been used? Maybe you can give people some context of how novel this approach is.
So it was done for several genes before, like in muscular dystrophies and in retinal disorders. And it's not too commonly used in translation so far. I think it's the first time this was really used in translation, this dual AAV for the OTOF gene therapy. But in preclinical, it was pretty common. There were some groups that systematically studied this kind of gene therapy vector systems using 2 or even 3 AAVs where the coding sequence is split to.
Wow. You've brought up the otoferlin gene a few times. I'd love if you could give a little bit of a high-level breakdown of, if we focus on early onset, childhood, infancy, or a little bit later onset hearing loss. When a child with that phenotype shows up in the clinic and you do genome sequencing, otoferlin is one of the genes that frequently comes up. But what's the broader universe? How many cases are gene negative? What are some of the other genes that we're looking at? I want to just help set the table a little bit to understand when we talk about the breakthroughs in otoferlin, what else there is and what some of the challenges are in the rest of the universe.
Yeah. So in first glance, it's not so obvious why among more than 150 different genes, we know the mutations of which cause hearing impairment. 3 companies and 2 research groups started to do gene therapy for OTOF gene. So what is so special about it is the children with biallelic mutations in OTOF are born profoundly deaf, but you can still record the outer hair cell function. So you have in the inner ear the inner hair cells, which are the true sensory cells. So they make the signal towards the brain, but you have outer hair cells which amplify the signal. And there's a rather easy and fast test to find if the outer hair cells are working. And this is used, for example, in newborn hearing screenings in most clinics. And in case of this form of deafness, the outer hair cell function is intact, but still no information is transported to the brain. So it typically takes a while until these children are diagnosed. So the parents get the result from the newborn screening, which says the inner ear is okay, the amplifier works, but then they observe that their child is just not reacting to any sound. It's not starting to speak. It is not a very common form of hearing impairment. So among those with recessive hearing inheritance and profound prelingual deafness, it is making up maybe 2% of the cases.
Oh, wow. And so why is that particular case so attractive for drug development?
We learned from mouse models, from otoferlin knockout mouse models, that the hair cells are still present. So they are not working, but you can see them morphologically. You can also see that the synapses are there. And we know that just the release of neurotransmitter from these inner hair cells is impaired. And we can measure that very nicely. So we kind of have an almost exact idea of what's going wrong, but the structures are intact. And they remain intact for months in mice. So that's why we were very optimistic that this form of gene therapy can be treated in humans postnatally, because there are a lot more gene therapy trials done in animal models, and in mice, the inner ear develops partially after birth. That means you can do a gene therapy in mice very early after birth and it rescues hearing. But if you do the gene therapy later, it does not rescue hearing anymore. And that would mean that in humans, you would have to do this gene therapy very early in pregnancy. And of course, this is nothing which is clinically easy to achieve. And that's why everybody looked for a candidate where you can presumably do this postnatally in humans. And OTOF is kind of the first candidate, because also it's not a degenerative disorder. I mean, if, for example, retinal disorders that are degenerative, you have to do the gene therapy before the degeneration starts. Right. So you also run the risk of destroying something which is functional. And in case of OTOF, the children are profoundly deaf. So there's hardly anything that can go wrong. So even if the gene therapy doesn't work, you would give the children a cochlear implant and they could start learning language. So there's a low risk, high gain potentially. And we were all confident that this would be doable after birth in children.
Are there other genes that share the same characteristic as OTOF where you could treat postnatally? And if so, I would love to hear your thoughts on what the potential next wave of opportunities could be.
This is a very hard one. So if there would be an equally good candidate, then it would be probably as advanced as the OTOF gene therapy. So the answer is there's no equally good candidate. So the next candidates are either those where you have a degenerative disorder, and for this, it is very helpful if we have now the results from the OTOF gene therapy, of which we, of course, learn what works well, what works better. And this knowledge will be really crucial to develop gene therapies for those hearing disorders that are progressive. Then there are some very, very rare ones. And the question is, will you find enough patients to do a clinical trial? And there's, for example, the most frequent form of hearing impairment. The gene is called GJB2. It's encoding for a connexin 26 protein. And about 1/4 of the children that are born hearing impaired have a genetic defect in this gene. So we have a common founder in the European population for this.
Wow.
1 out of 50 is a carrier for this mutation. That's why it's so frequent. There are occasionally children that are born with only moderate hearing impairment, and we hope now that with a gene therapy, for those the hearing can be rescued. But most of the children, I have to admit, are born really with profound deafness, and for this, I personally don't think that the gene therapy will rescue much, but maybe we can help those that are born with some residual hearing. But this is still in development. It might be in clinical trials, maybe 2 to 5 years from now.
Yeah, that makes sense. And with that one, if we linger on GJB2 for a second, is that because there's some kind of structural damage or lack of development in one of these crucial hair cells that you can't rescue, basically? Maybe you could talk a little bit more about why that would be so hard to reverse.
There's a lack in good animal models. This is the only gene where the defect is having a worse phenotype in mice than in humans, because in mice it's required for some glucose transport in the placenta. That means the knockout is embryonic lethal. And that's why it is very tricky to find a mouse model. You have to take a conditional mouse model, but at the same time, the gene is expressed in many different cell types, and it's very hard to make a conditional knockout that really reflects the situation in the humans, because as soon as you have not targeted one type of cell, then you don't know for sure if this really recapitulates the human phenotype. So that's a big challenge, and that's why the therapy for this one is lacking very much behind. And the second reason is, from the mouse models we know that there's pretty big structural damage to the inner ear for those that are born with hearing impairment. We assume, therefore, that there's not much we can do with gene therapy.
Yeah. This is quite a conundrum how you go after these, but the fact that you mentioned 1 in 50 people are carriers for this in many European countries, I imagine, has me thinking about carrier screening. How much do you think about carrier screening or newborn screening in cases like this as a potentially a different route to think about prevention?
Yeah, I think carrier screening is more straightforward than prenatal gene therapy, for sure. Because if you know some relatives, or if you're really afraid of having a hearing impaired child, I would rather go for carrier screening than doing a kind of genetic analysis in the first trimester to have enough time for a gene therapy, which needs to be done before 18th week of gestation. So either you accept that your child might be born with hearing impairment, there's still always some risk, but we have, with cochlear implants, we have a good therapeutic option for all of them, which is maybe not as good as gene therapy, but it's still a very valuable tool. So the children can mostly go to normal schools. They need only a little bit of support in daily life. And so this is always a good option. So I personally would not go for a carrier screening, but if this is really what you fear, you could do it.
Yeah. With this potential possibility in the future of gene therapy in 18 weeks of gestation, I think that you said, do you see this being a path that the field gets to in the next 10 years, say, or is this really far in the future? Because the lack of mouse model and just the challenges with having to do something this intense and novel in that kind of environment seems challenging to me, but it may be closer than I realize. I'd love to get your thoughts on how far or close we are from considering that.
That's a very interesting question. So I personally don't think it will come in the next 10 years, also because this is a quite invasive procedure and there's a high risk of losing the fetus if you would do that.
Yeah.
So, and of course, you need to do the diagnostics beforehand. So we know that you can do some genetic analysis from the blood of the mother. So maybe these tools will be sensitive enough to detect a homozygous mutation in the future. But still, I mean, I could not imagine. So I'm a mother of children. I could not imagine to have such an invasive procedure so early in pregnancy with quite some risk associated to it. So I think pre-implantation diagnostics would be a more straightforward way to go than gene therapy before 18th week of gestation. But that's my personal opinion.
Yeah, that makes sense. What about more gene agnostic approaches, or at least broader approaches to treating deafness or hearing impairment? What is your work understanding some of the common mechanisms that underpin multiple of these genes tell us about the possibility for going upstream in some way to restore, maybe not completely restore function, but at least make some progress? Is there anything that you've learned about that?
So we are still in the phase where we look at genes one by one and we try to understand the particular pathomechanism. We try to look up the expression in which cells is this gene expressed, is the expression in these cells essential or not. So we are still going one by one, but I think this has some more potential with respect to age-related or noise-induced hearing loss. So we are beginning to understand some common mechanisms there, and I have some hope that in the future we develop more and more ideas with which we could protect the inner ear or enhance endogenous protective mechanisms. And this would be something that would be applicable to many people, like those that have a noise exposure or those that have a higher risk of age-related hearing loss, or maybe even to all people, because eventually we become old and we will get hearing impaired.
Yeah, I'd love if you could talk a little bit more about that. What ideas do you have in that direction?
As I said, so we are trying to understand endogenous protective mechanisms, but we are just at the beginning of that. So for example, some groups found out that estrogen has some protective mechanisms in women, and the diabetic drug metformin can help in male mice at least. So we are just at the beginning of dissecting such protective or regenerative pathways, and I think we are still too far away to have a really straightforward idea with which we could help all people. It will take some more basic research to understand more about that, but for the future, I'm quite optimistic that we will find something.
Yeah, I have a very simple model in my head of how age-related hearing loss works, which is that we all get exposure to different levels of noise throughout our lives based on our occupation or how loud we crank those AirPods up. But we also have a genetic background that may influence how quickly or slowly that external influence takes root. What I don't have a really good mental model on is how that genetic influence relates to the environmental one. Like, what is it exactly that you can be genetically predisposed to? Are there structural differences in the inner ear? Or is it to do with our, endogenous repair mechanisms of how we rejuvenate these cells over time? Any info you can share about how this like push and pull of the environment and our genetics, what our understanding of that today, I think would be really interesting.
We are still lacking a systematic analysis of that. I wanted to do that. I'm still trying to find money for that. So we don't have really a good idea. So we know that the immune system is involved, some inflammatory responses after a noise, some specific immune cells help with the repair, some do more of inflammation and make the problem even worse. So, but this is just a snapshot, just a little piece of knowledge we have around the whole system, and we have not really a good idea on which genes are involved there. I'm also not a specialist on that, so there are other people doing genome-wide association studies to find out which genes are associated with more or less hearing impairment in aging.
What are the big focus points of your lab right now? If you could share a little bit with people about what you're working on today and what's got you most excited going forward.
We are actually indeed working on 2 more deafness genes, doing basic research, trying to understand the pathomechanism. And then once we understood this, we can go ahead with developing a targeted therapy. But we have also projects where we look at signaling pathways, and we think that these are also involved in the protection after a noise trauma. And this is what we are currently studying in animal models.
Yes, with those 2 genes, I don't know if you can say which ones they are, but if you can't, just having an understanding of what the biggest challenges are that you're trying to unpick. I know you do a lot of mouse work, so you have to start with a good mouse model. Is it about timing of delivery? Is it about what the actual therapeutic modality could be? Because we talked a lot about gene therapy, but there are other options you can consider, right? Depending whether it's recessive, dominant. Would love to get your sense of what the frontier is, and as you're looking at the next gene, the second or the third that we could go after. What are the big challenges that you have to overcome?
So it's in both cases degenerative disorders, and we need to understand what precedes the degeneration of the cells. So we need to understand which mechanisms are behind the cell death and where we have to step in to protect from cell death. That's currently the challenges we are facing. And it's not so obvious. So we have one gene, the TMPRSS3 gene, which causes hearing impairment. And this is known that the first neurons in the auditory pathway, the spiral ganglion neurons, degenerate over time. But now we just found out that the mature spiral ganglion neurons do not express this gene at all. So we need to find out why the cells degenerate, although they are not expressing this gene. So we think that there's some signaling from other cells that express it, but this is something we have to study still.
Is the mouse model generally the best method that we have? Are there any good organoids or other systems, or is as imperfect as it is, the mouse is, or do you have to triangulate, I guess, from a few different directions sometimes?
Yeah, interesting. Exactly for this project, we are using both mouse models and organoids from human cells. And I think they are quite comprehensive to each other. And so we can study if the mechanisms are similar in human cells as we see them in mice, but the cells in the mice are much more mature than what we can reach in the organoids. So we have still a kind of immature cell types in the organoids, which is interesting if you want to study something which takes place during the development.
Yeah.
But for finally, like for degeneration, this is not a good model.
Yeah, that makes sense. Are there tools in the toolbox that you're missing that you wish you had to better understand? Like, is there any black box of information right now that you really don't have because you don't have the right tools?
Not really. So for most genes, the mouse models are really excellent models. The mouse inner ear works really, really similar to the human ear. And with the exception of the GJB2 gene, which we talked before, the pathomechanism is very much alike in the mice and in the humans. So only the speed of progression for progressive forms of hearing impairment, this can be a bit different in the mouse models. But apart from that, so we can really translate that one-to-one from the mice to the humans.
Yeah. I'm curious to get your perspective on the therapeutic side, if there are any things that you're particularly excited about. You've worked on, sounds like dual AAV and some of the more frontier technologies already. Is there anything that's up and coming that you think is interesting as it pertains to genetic hearing impairment or more broadly?
That's an interesting question. So there are some new tools that came up, like gene editing, prime editing, things like that. They are currently very much hyped, but I see the problem that you have to design them specifically for each mutation. And for example, in the OTOF gene, we know more than 200 mutations. Right. So I personally favor the one-fits-all approaches. There's one interesting approach called HITI, where you insert the coding sequence in the first intron, so you can overcome all the mutations that are in exons 2 to whatever. and it still uses the endogenous promoter mostly. I find this quite interesting, and unless you have mutations in the first exon, you can treat all other mutations with the same approach. And this would also be useful for those genes that need a very specific expression pattern because you make use of the endogenous promoter. This is not such a problem for OTOF. We need a really high expression for otoferlin in the cells. This cannot be high enough. But some other genes, if you express them too high in the cells, this causes problems. And for this other approach would be very interesting.
Yeah, really interesting. I want to ask you about your career trajectory as well. My understanding is that you started by studying biochemistry in Tübingen, where you are today. You took a detour in lots of other places, but now you're actually back very close to where it all began. I'd love to just get a little bit of a sense of how that career trajectory has been, and that's not so common that people go back to where they originally studied. So I'm just curious to hear how that came about.
Yeah, that was more a coincidence. So as you said, I studied biochemistry in Tübingen. Then I started my PhD in Tübingen, but my PhD supervisor moved to Freiburg. So I moved with him to Freiburg. And then the group of Tobias Moser hired me in Göttingen, which is a very famous group for inner ear biology, one of the top leading labs for inner ear basic research. And I went to Göttingen and I got the chance of starting my own first junior research group and later research group. And I could do my own projects, like, for example, this gene therapy for OTOF project. And then with looking for permanent positions, my mentor, Tobias Moser, had the idea of asking some newly appointed head of an ENT department if he or she would be interested in my research. And just by chance, that it was not published at that time that I had this data on the gene therapy for OTOF. The newly appointed head of the ENT department in Tübingen here, Hubert Löwenheim, he said he would be interested in hiring someone who does gene therapy for hearing impairment. And I acquired some starting money. This is called Heisenberg program from the German Research Foundation. And with that, it was quite easy to get hired here in Tübingen on a permanent position.
That's very cool. You mentioned the group at Göttingen that was prolific in the field and that you spent a lot of time in. What was it that made that group so impactful?
So Tobias Moser is a genius head of this group. So he has a really deep understanding of a lot of mechanisms. He's one of the pioneers for patch clamp electrophysiology of the inner ear. So there's a lot of techniques established in this lab, and he's very open towards new methods, new techniques, and he's also very good at acquiring money, and he has a large group and keeps all the people together. So that makes this group very successful.
Are there any mysteries of the inner ear today that we don't understand? It's this tiny part of the body, but it's amazing and exquisite and does all these fascinating things. But it strikes me as it might be kind of hard to study in humans outside of cases where it goes wrong. But researchers are very creative. Is there anything that we really don't know fundamental about how the inner ear works, or do we have a pretty good understanding?
I think we have pretty good understanding of the basics. So, but there's, of course, always questions that are not yet answered. For example, what is striking is that normal neurons, if they do neurotransmitter release, they use the so-called snare proteins for vesicle exocytosis, SNAP25, synaptobrevin, syntaxin, and tagmin. And we found out, and others found out, that the hair cells of the inner ear do not use these proteins for exocytosis. They use otoferlin, this is known, and we think they are also using other proteins that form a machinery for exocytosis in inner hair cells, but still these other proteins are not known. So we are not entirely sure if otoferlin is the only protein that is required for triggering calcium-induced exocytosis in these cells, or if there's more proteins we still don't know.
Yeah, interesting. That is a great answer to my question. So I was also curious to get your initial thoughts on what impact the gene therapies that we're seeing today are having on these kids that are being treated, because the alternative is a cochlear implant, which does restore hearing in some way, right? And so the big question on my mind, and probably on many people in the field's mind, is what is the difference, and how does this trend over time from a gene transplant, gene therapy type of approach that in theory could restore hearing in a more fundamental way to the medical device that we have today that most kids that have this condition have the option of taking on?
Yeah, that's a very interesting question. So the cochlear implants are very well established in the lab. There are more than 1 million users worldwide, and for most of them, it works pretty fine, particularly for language acquisition, speech comprehension. It is not as good for pitch perception, so for music. So because you can't distinguish frequencies that are closer than 1 octave apart. So it's not a good thing for music. Although some people have a good pitch perception, but we don't understand entirely why it works in maybe 10% of the people better, but in 90% of the people, it's not as good. So we think that the gene therapy, of course, would be much better in pitch perception. But we also know from patients that have low otoferlin protein levels due to point mutations in otoferlin that they have problems in speech comprehension, particularly in background noise. And they might even have an auditory fatigue, which means that if you present them a constant tone, they perceive this as fading out. So we cannot measure directly how good the otoferlin levels are in the children that have received the gene therapy. So we don't know really for sure how good their speech perception will be. So most children were very young when they were treated, and we have to wait 3, 4, 5 years until we can really make accurate measurements of the consonant recognition, for example. But this is a very interesting point I'm thinking about. It will also make a difference which language you're using. So some of the European languages, like Italian, for example, they have a lot of consonants. And we have some data from the mouse telling that if you have too low otoferlin levels, so if the gene therapy is not raising the levels to normal, then you have problems in consonant recognition. And the consonant recognition in the cochlear implants is also not 100%, but it might be better. And now if you think, for example, of the Chinese language, which uses hardly any consonants, but you need the pitch perception, you need the prosody. So if the syllable pitch goes up or down, this is a very important information. The Chinese language has only about 400 different syllables, and it's very crucial that you understand this tone of the speech. So I think personally that for Chinese, the gene therapy will be superior over cochlear implant, even if the gene therapy is not ideal. But there can be languages where you don't need pitch perception, but you need consonant recognition, where maybe cochlear implant will be better. And for this, I think what we can do now is to treat 1 ear with gene therapy and the other with cochlear implant. Then we have the best of both worlds. But that's just a recommendation. Some people are more optimistic about the gene therapy, that it can be better than the cochlear implant, even for those languages with high consonants.
How would you actually measure that over time? I guess we have the benefit of there being millions of people, like you mentioned, that have cochlear implants, but this strikes me as a tricky thing to measure.
It is definitively, and it takes years until the language acquisition in those children is as good that you can really make these accurate tests like consonant recognition, monosyllable, disyllable recognition, sentence recognitions. That is what is typically done. But of course, it's different from language to language. For example, in the Greek language, you don't have monosyllables for whatever strange reasons. So you can't compare from Greek, which is the monosyllable percentage of recognition. So we still have to be a bit careful about that. And also the different approaches might lead to differences in otoferlin levels. And even from child to child, this can be different. What we can see maybe from the amplitudes of the brainstem responses, we can draw maybe some conclusions in the future, but also this undergoes some developmental change. So we have to see if this is a good measure on judging how good the hearing is in the children.
Fascinating. This has been an amazing conversation, Ellen. Thank you. Is there anything that you want to leave people with before we close out?
Not for now. I'm pretty happy.
Great. Well, thank you for your time, and thanks everyone for listening as always, and we'll see you next time.
Thanks, as always, for tuning in to the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.